Good afternoon, everyone. I know I'm standing between you and coffee, which is a very dangerous place to be. I can read it from your faces. But I try to keep it short and engaging. And if anybody is dozing off, I just assume that you are doing meditation on my slides. So here I'm going to talk about feasibility study on the synthesis of trimer-based DNA sequences for combinatorial DNA encoding in DNA storage. First, I want to talk a little bit about our synthesis method, which is photolithography. This is our machine. This is we illuminating UV on a glass slide, and thank you. Yeah, and on the glass slide, we are synthesizing DNA by delivering chemical material into a flow cell where the chemical goes, and the UV light can do the rest. And this is the illumination system, which I will explain a little bit more. And this is the synthesis style. Also, I will explain it a little bit in detail. So the heart of our synthesis system is a digital micromirror device, which is made of more than 2 million controllable mirrors. As you see the size of the mirrors, each of them can be turned on and off. And the UV light can go through a few lenses and a few mirrors until it can project an image that is here, project that image onto this flow cell, which our glass slide will be placed, and the synthesis will be occurred here. We can synthesize two different formats, let's say, which when we are going to synthesize in higher density, we can synthesize more than 2 million sequences, 2 million different sequences at the same time on one slide. And this is the cycle of the synthesis. Basically, we are using phosphoramidites, which made us enabled to do the synthesis. Phosphoramidites are, let's say, simply just some nucleotides which have a lock on the top, which has a cap on the top. And that lock, which is shown as a sphere here, can be deprotected or unlocked or removed by UV light. And this is the part where UV can work. So imagine at the surface, at the first round, we have this nucleotide, which are all having its own lock. So if we are turning on the UV light at this position and at this position, those locks are unlocked, those caps are removed, and the hydroxyl group at those parts are available for the next round. So if we are adding, for example, G, the nucleotide of G, at the next round, G will be sit only in those places that are available. So in this case, for example, at the next round, we are unlocking these two parts and adding T, and this T only plays at this position. And in this case, we are just sequentially can synthesize different sequences at the same time on the glass slide. So, but we are here. I just showed you the synthesis with monomers, but my synthesis that I'm going to talk about is synthesis with trimers. What is trimers first? So imagine instead of monomers, well, we are buying, I'm just saying this for those who are not really familiar with chemistry, we are just buying A, C, G, T in different bottles from the market and use it for the synthesis. It is as simple as that. But imagine instead of buying monomers, we can buy trimers as the synthesis unit. So we have a bottle, for example, that only have AAT in it as a unit. So when we are using them as the unit for the synthesis, AAT will be sit in those places that are available and there is not, the error rate, it is almost impossible in between. So imagine we have these 16 different trimers, and instead of using only one unit at each step, we are going to use 5 different trimers at the same time and introduce it to each location. So each location, I mean here that I showed you. So at this position that G is set here, imagine instead of G, it's a mixture of G and C. So like 50%, 50%. And when we are reading the data from the sequencing, in that position, we can see C and G 50% and 50%. So in this case, we can interpret that ratio as a new letter. So we are introducing a new letter when we are converting digital data into DNA. So in this case, if we are mixing C and G or G and A and T and A, we are just adding like 6 to, I guess, 15 more letters. But if we are using, we are picking 5 out of this mixture, it's a combination mathematics. It is 16 over 5. We can introduce more than 4,000 more letters. In this case, when we are converting digital data into DNA, we can convert each 12 bits into 1 letter, and in the end, around exactly 4,096 letters are available for us when we are converting the data from digital data into DNA. So it makes it way more intense when we are storing the data. So let's go to the result. So at first, I want to tell you that the one problem with the trimers are that they are not having the common lock as monomers. The monomers that we're using, they were phosphoramidites. They will be unlocked. They can be unlocked with UV. But for now, the trimers cannot, they are not available in that form. They are only unlocked with acid. So at first, I had to do some test synthesis and optimize the synthesis of phosphoramidite that can be unlocked with UV. At the same time, at the same sequence with the phosphoramidite and trimers that can be unlocked with acid. So the photo is not really clear, but after doing so many optimization syntheses with changing different factors in the synthesis, it's a really multi-step factor. I reached to a position that I could really have a good signal to noise ratio, and I just used that method, that protocol for further for the synthesis of trimers. Also, in this case, I used a few nucleotides that are locked by that can be unlocked by acid, with nucleotide that can be unlocked with UV in one sequence. So as you see, in A we have 4 nucleotides that unlocked by acid, which is this one. This is a fluorescence. The green part is a fluorescence light that you see. And at the B sequence, we have 6 nucleotides in the sequence that can be unlocked by UV in the mixture of the other common nucleotide. And the C is fully nucleotide phosphoramidites that unlocked by UV. And we can see almost they are having the same signal to noise ratio. Also, in this image, also I used way more number of nucleotides that are unlocked by acid in the sequence. We can say almost they are the same, but a little bit, the fluorescence reduced a little bit, which is normal. After the optimization and proving that it can work and we can use these different kind of phosphoramidites, this kind of units at the same time in one sequence, we went for the synthesis of trimers. The trimer that I used is this. So it is one molecule, one unit, consisting T, A, and C. So this is my sequence. As you see, all of them that are in ACGT are phosphoramidites that are unlocked by UV. But number 6 is the trimer. And I use number 6 because the 6 is readable by our chemical delivery machine. So in the first sequence, I just add one trimer in this part. The second sequence, this is 2 trimer consequentially added. And the third sequence, 2 trimer consequentially added and also separately with another trimer. And the last one is just a control. Working? Where is the computer? No. Okay. This is the first sequence that I showed you. I hope the laser works. Yes. So this TAC that you see here in bold, this is the trimer that I added in the synthesis. And as you see, you can see that trimer is well attached to the sequences in the mix. And we had a very good result in this variant at the first sequence. And this result is very promising for us because in the end, we are going to use a mixture of these trimers. This is not the final step. This is just proof of concept that we can use the trimer in the sequence. Also, 2 consequential trimers also worked. As you see here, this TAC and this TAC are in the sequence in the mixture of the other monomers. And it also works, also a little bit less efficiency. And also this one, as you see, we have so much error when we are using 2 trimers consequentially separated from another one. As you see here, we have a lot of errors here, and this sequence didn't work very well, which we are working on it to make it work. But this is only the basis until we can use the mixture and as a proof of concept using the combinatorial letters. The other things that I really wanted to bring the result here, but unfortunately we couldn't get the result yet from the sequencing, is that in the experiment that we've done, this time we used 3 different trimers, TAC, TCT, and CTT at the same time. In the other experiment, we only used 1 trimer. And in this case, we are going to use the mixture we did this experiment, the mixture of 2 trimers in different ones, X1, X2, X1, X3, X2, X3, and also a mixture of X1, X2, and X3. So hopefully for the next conference, I can bring this result. And in the end, I want to thank you, my colleagues, and if you have any questions before coffee, I'm available.
Thank you, Hamed. Questions? Yes, we see. Perfect timing.
Coffee is important.
Thank you for the interesting talk. I am curious about how difficult it is to make the trimers, and if you see that process being scalable in place of, just the single amidites.
Well, honestly speaking, it is just a proof of concept, and money-wise speaking, it's not it's not efficient, because trimers are very expensive, and when we are if we are buying monomers, they are very cheap, they are very available, but buying trimers first it's very difficult, and then it's very expensive. The trimers that I bought here, I could use only for 2-3 rounds at most, and it cost me a lot. And for this experiment, for example, only for one experiment, it cost me around 2,000 euros for one experiment. So for now, it is a proof of concept for the combinatorial letter that we proved that it's working. But currently, it's not. I guess it's not feasible money-wise. Yeah.
Thank you. Then I have a question. So if you say it's not feasible money-wise, so what is the end goal of the experiment? Like, why are you doing this?
For sure, there are a lot of things. Also, the machine that we are developing is costing, the one that we just sold, it costed 100,000 euros. So everything is expensive. But for example, for the next machine that we are we are trying to make, it is way cheaper. So hopefully the people who are making trimers, because we are not making trimers, we are just buying them, hopefully they can make the workflow more efficient and make it cheaper, way cheaper, yeah.
So what potential do you see in the trimers themselves that you're trying to?
The trimer, as I explained, it increases the density of the data that we can store here. Yeah. So it adds, for example, if I use ACGT, we only have 4 letters. But if we use this method, we are having more than 4,000 letters, and it increases the density of storing data.
Thank you. Yes, question.
I have a related question. We have seen a lot of discussion about error correction codes with only 4 letters that are not distributed as percentage of letters. So apart from doing experiment, did you simulate how errors within insertion, deletion, substitution would impact this strategy of using different percentage of these trimers?
Well, I am at the chemical part of this experiment. The idea is from this data from Zohar Yakhini, and we are collaborating with them. They developed a workflow for now just analyzing the data from trimers, so it can read the trimers as a unit, not as monomers, because when we are sequencing it, we cannot see any difference. It is just like monomers. We cannot see any difference. So they are working on it, that when we are analyzing it, they got the trimers as monomers. And if there is some insertion or deletion at the trimer, somehow they ignore it, because there shouldn't be any deletion or insertion there. And if there is any, it is not from the synthesis. It is added at the PCR for the library preparation, and also in the sequencing part. Yeah.
Yeah, but I mean, when we are reading a sequence, we expect only one signal at each letter. And in your case, you would have a mixture of signals. So I guess this mixture would impact on the error rate.
We talked about it. That's very important. We are adding specific letters, monomers, before and after the trimers. So when we read that signal, we knew that after that it's trimers. So somehow we read it as a unit, something like that. But I'm not that expert in that.
Yeah, that's a good trick to mitigate errors. Yeah. Thank you.
Yeah.
Okay, last one.
Hello, Hamid. Thank you. I have a question. What's the minimal amount of molecules that you can or that you synthesize per spots? You talked about high density.
Yes. Well, I said in the density, I said it is the density of information in one sequence. but also that density is also meaningful here. When I said 2 million different sequences at the same time in one slide, it doesn't mean that literally 2 million sequences, 2 million kinds of sequence. For example, at each of these mirrors that are, the UV light is emitted and projected on the glass slide, at each of these points that you see, one kind of sequence will be synthesized. So the size of each of them is roughly 10 square micrometer. So just you can calculate how many DNA can be synthesized at the same time in only one part.
Do you know, Erhard?
I calculated once, to be honest. So if we consider a DNA as a cylinder, we can calculate that number, but I guess it's roughly 1 million. But I'm not sure. I calculated once, yeah. So 1 million sequence of the same sequence at each of these parts.
Thank you for the questions. I really hope Hamed will become very popular during the coffee break, as you have a lot of questions for him. So thank you, Hamed, once more.
You're welcome.